So I'm really excited to have Dr. Clare Salzler here today, and we worked together on the NIP study, the Nutritional Intervention to Prevent Diabetes, along with Dr. Peter Chase, and we have a long history together.
Yes,
so
we do.
anyhow, he calls me his PI, Principal Instigator. So Dr. Clare Salzler is the professor and chair of experimental pathology at the University of Florida at Gainesville. He's been very involved in PANDA, the DPT-1 study, and he's also the director of the Center for Immunology and Transplantation, and he's the Stetson Professor in Experimental Pathology, and he also sees a lot of patients. And so there's an expression, if you want to get something done, you give it to the busiest person in the room. And both of these guys here qualify for that. So the story on how I met Dr. Clare Salzler is we were on the JDRF Grant Review Committee. I was on there for about 6 years, and I read this grant, and I came back to Dr. Peter Chase, who is my mentor, and I said, I just read the best grant that JDRF has ever looked at. And I was so excited about it, and it was about the use of fish oil in people to reverse type 1 diabetes. Or in at least the mouse model, and he wanted to try it in humans. So I didn't know it at the time, but shortly thereafter, I got a phone call from Dr. Revers at the Barbara Davis Center, who's running the DAISY study, and he said, I don't want you to worry, but I have one son who was diagnosed at age 1. He said, don't worry though, with your son Ben, don't worry. He has at least 2 months before he's going to be diagnosed. So I hung up the phone, called United Airlines, and was at Dr. Clare Salzler's lab the next day, having been drawn to look at cyclooxygenase 2 levels, because back then you had to use whole blood, warm blood. And he went on fish oil. And you guys might know the story, and some of you do, but I think it was about 5 months later he was retested and his autoantibodies had reversed. And Dr. Sears makes excellent points about finding something that you can do for the rest of your life to decrease inflammation and to activate and regenerate cells, because when he's gone off of it, he's become autoantibody positive again. And then he goes back on his fish oil, and he has become his own experiment, not by my choice, but even recently got ZNT8, went back on his fish oil, and that reversed again. So for people who say it's inevitable if you have 2 or more autoantibodies, I think Dr. Clare Salzler has some optimism that might not be true. So anyways, thank you for all you've done.
Thanks. Well, thanks, Sonia and Camille, for the invitation here. And what I want to present today is some of the work we've been doing in the lab recently. And this is the way I look at diabetes and understanding pathogenesis is a lot like trying to understand a play. It usually comes in pieces, acts 1 through 4 if you're a Shakespeare fan for most part, and but it's a lot like watching Game of Thrones though. We've watched seasons 1 through 7. We got a good understanding of all the characters, but we have no idea what's going to happen in the last season 8. So we have all this information. We continue to build on it, and we try to, I guess, look forward to the ways to prevent disease. And a lot of people have been working in this field for decades, and we've made progress, no doubt. But I think we're still moving towards a complete understanding, and probably subset understanding of patients with type 1 disease. Now, one of the things that's really limited us has been the capacity to study the process going forward in chronic inflammation. So most of the disease models that we look at in vitro in the laboratory, whether it's gene expression or protein expression, cytokine expression, are snapshots of a single moment in time for the most part. But this is what we're dealing with, is systems biology at its most challenging, and trying to understand the process but also understanding the process in the continuum of time, and also some complicating factors such as environment and so on. We'll talk a little bit about viruses today. So it's a very daunting task. So what I'm going to try to do today is to kind of work through a bit of a current snapshot we have of what we consider a very much a longitudinal process in understanding each of those segments of that process to get a better idea of when to intervene and with what and moving forward. But underlying all this is inflammation, chronic inflammation, and omega-3s and vitamin Ds are very, very essential to, as Barry put it, resolving the inflammation that occurs. So I have no conflicts. I had a consultation with Neobax regarding some anti-interferon approaches, which we haven't had any further conversation, so that's kind of on hold. So these are the players that we all know, which are the environment, which includes nutrition, and in this case, DHA, EPA, and vitamin D being the ones that we're talking about today, and there are potentially others. And the other is the world of microbiology, particularly viruses, and there's many different types of viruses, but we have a number that we've studied, and Coxsackie virus is one of those that is potentially the most studied. And the genetics of disease, there's 40, 50, 60 susceptibility genes that we are looking at and dealing with. And what I'm going to focus on today are those that are involved in either the response to viruses or to interferon signaling pathways, which follow viral recognition in tissues and in immune cells and lead to activation, in this case chronic activation over a period of time. And then there's the target tissue, the beta cell, and then the effector system, the immune response, that largely leads to the development of disease. And one cell type that we'll talk about a little bit are plasmacytoid dendritic cells, which are the producers of interferons within the human body. So all this conspires, if you will, to the development of beta cell destruction or dysfunction, and to immune activation and modulation, which includes a whole cast of characters, including T cells and antigen presenting cells, as Camille referred to. And then a new set of cells which are currently being looked at in the lab for stem memory T cells, which may end up being the repository for autoreactivity, as well as the phenomenon of exhaustion in T cells. So this is the continuum, and I won't go through the entire list of this, but we all know that there's probably a phase of pre-type 1 diabetes, which is individuals who have genetic predisposition. They have a family history that obviously may align the genetic susceptibility genes that will allow a person to have a risk for diabetes. And then there's changes in the immune system, nutrition, etc., that happen as an effect of our growing up or aging, and that modifies the immune response as well as how we respond to it. The nutritional factors that we take in that may help to contain or calm down inflammation, and the inflammatory response, which in patients with type 1 diabetes, as in lupus as well, are accelerated and accentuated, and that's largely upon the genetic polymorphisms that exist in some of the genes that we'll talk about. And then there may be, we have data from the NOD mice that suggests that these stem memory T cells are present in the NOD, and they develop during gestation. So in utero, there's the evolution of a population of autoreactive T cells that can be ablated by treatments with anti-interferon antibodies. And then, so we progress over time to the point where we can detect autoimmunity by primarily antibody detection. And at that time, people have looked at viruses, including the Coxsackie virus, and as we'll talk about more, interferons families, type 1 and 3. The activation of the autoimmune response becomes activated during this time, and there's an expansion of T stem memory cells, as we'll show you. and then the development of beta cell dysfunction or destruction. And the question is, when it comes to the virus and the pathogenesis of type 1 diabetes, which came first? Did we have in sight autoimmune responses in progress, and did viruses come along and accelerate that or accentuate the response? Or did the presence of viral infection of beta cells lead to the activation of autoimmune responses and progression to diabetes? And then there's, I think, a neglected area of type 1 diabetes pathogenesis, which is established patients. And the reason I say it's neglected and potentially important is that in the NPOD studies that have been done by many people, there's a presence of beta cells that have insulin. Now, they're reduced in number. The level of insulin expression may be limited, but those beta cells do not function normally. They don't release insulin in response to glucose. And so there's, I think, an opportunity there, if we can ever master beta cell regeneration, to understand how to resuscitate that function of beta cells and potentially provide another approach to this disease process in patients with established disease. So what we're going to start out doing is talking about type 1 and type 1 interferons and type 1 diabetes. And there's a number of human studies that have supported the role of these cytokines in diabetes, lupus, rheumatoid arthritis, others as well. And in the old days, when we used to treat hepatitis C with interferons, that there were a number of patients that, as a consequence of this treatment, developed autoimmune diseases, including type 1, lupus, and others. There's also been some studies done by various investigators that have looked at the expression of these cytokines in the blood of individuals using ELISA assays primarily, as well as the inflamed islets as well. There's a report of a blush of interferon detection and maybe gene expression before the appearance of type 1 diabetes, and we'll talk a little bit about what we've done in that area. So first of all, to get into is the interferon response. So how does one respond to a specific cytokine? And there are polymorphisms in genes that control the signaling pathways for interferons, much as they do with other cytokine gene receptor signaling pathways. And in type 1 diabetes, the IFIH1 gene, which encodes the MDA5 sensor for viruses, particularly double-stranded RNA viruses or intermediates, as well as a principal and important proximal signaling factor, TYK2, which helps to translate after the binding of the cytokine to its receptor on the cell surface, denoted here in that gray little guy up here, that TYK2 right here is primarily in a position to accelerate that. Now there's polymorphisms that create enhanced function and limited function, and interestingly those that are associated with a lower response to interferons are those that are protective for disease. So we can go through this list of factors that are associated with signaling here, and those that downregulate, generally downregulate the interferon response, and the polymorphisms that do that. If you have the form of the gene that causes enhanced function, then you're going to develop you have a higher risk for disease. So it's an inverse relationship between the signaling effects and the polymorphisms which regulate those potencies. So in this case, we've kind of put the blue arrows here up next to some of those factors which control the signaling. So we'll show you some data later that indicates that in type 1 diabetics, there's a heightened responsiveness in some individuals, and we'll show you the genotypes associated with that. So the next step is a Coxsackie virus. So viruses infect many cells. Coxsackie has a tendency to go to the blood, through the blood into the pancreas, causing pancreatitis or insulitis potentially, or to the brain in some cases, or heart. Enteroviruses are widespread in the environment. They're in water sources. They cause a lot of diseases outside the heart. Foot and mouth disease is particularly out in Southeast Asia. It's also an enterovirus that causes that. And there's some data, without going through the details of the studies, that suggest that these viruses maybe precipitate diabetes and have been shown to accelerate potentially the development of the disease when individuals are infected before, when they have autoantibodies. So over here, we have a picture from the NPOD collection that we did in the lab looking for interferons which are produced in response to viruses. And what we find here in green is the Coxsackie VP1 protein, which is a lot of folks have used in the past to demonstrate the presence of virus in the islets. And the purple cells here are beta cells that also express type 1 interferon, in this case interferon alpha. And then interferon alpha alone you would see as a cell with a red signature. So this suggests that there's virus infection, and this is an individual who had diabetes for 4 years. So these are persistent infections. We have data from some individuals, small numbers, that are at new onset, they have virus, and there's also some studies from others as well that show that it's present in autoantibody positive but non-diabetic individuals. So the survey that we've done in the collection at NPOD at UF shows, if we look at individuals with type 1 diabetes on the right and their duration of disease indicated here in the second to the last column, that these individuals maintain expression of the VP1 protein or in situ RNA expression, another way of looking at this, over several years after they've developed diabetes. Now this could mean they developed the infection after diabetes, or it could be that they developed it before. So we haven't answered that question there. But if you look in controls, there's only about 10% of patients, which is consistent with other studies, that you find in individuals who don't have diabetes. So this is pancreata from normal individuals with no history of diabetes. So suggesting that there's either a tendency for virus infection in diabetes or potentially an inability to clear a viral infection from the islets. So what about the expression of interferons in these infected islets? As we showed you in the first slide, there's a control islet here on the left, stained for interferon, which should show up as red, and we don't see interferon expression in control islets. The center is a patient that had diabetes, and this was after 12 years, 1 year of duration of diabetes, who died at age 12. And as you can see, there's a co-expression, the purple color of insulin and interferon alpha and beta in this case. And then there's an islet on the right which is showing staining for interferon alpha in the same way. So there's a coexistence of viral infection and interferon responses that we see in expression, I should say, in the islets of Langerhans. And this is a survey that we did with controls and patients, and then this is here, again, the percentage of islets. So that if you look between the controls and the patients, most of those have fewer islets, which is what we see in type 1. But the islets that are there are insulin positive, and they have also interferons expressed within those. We've also looked for type 3 interferon, interferon lambda, and that's likewise expressed there. It's another antiviral cytokine. And these are some data that we did in a beta cell line, which has its limitations, showing different forms of the virus, which are molecular mutants. They have a truncated 5' end, which slows viral replication and may be relevant to the development of diabetes. And this is the induction of type 1-3s, left and right, as well as a number of other cytokines. So the variance in the molecular structure of the virus can dictate the level of the cytokine that's produced. And it's not clear to us yet whether these truncated variants are really relevant to diabetes pathogenesis. But it's a nice way to explain why it's a slower and indolent infection. And also, the type 1 interferons, in this case the alpha and beta interferons here, interfere with glucose-stimulated insulin release when we do this study in vitro with isolated islets. Interferon gamma, which is type 2 interferons, and controls where we don't add any interferons in this case, do not suppress glucose-stimulated insulin secretion. So these cytokines have the capacity to potentiate destruction of the islets, but also to just inherently interfere with glucose sensing by the islets. Also looking at the capacity of individuals to produce interferons. We looked at plasmacytoid DCs few years ago, and we find higher numbers of these in patients with type 1 diabetes who are at risk with antibodies, new onsets, or established patients. So these are controls over here on the left, are at risk, antibody positives, new onsets, and established. And there's a significant increase in the number of these cells, particularly in the earlier phases of disease. There's also, we looked at different age cohorts, and unlike controls where PDC numbers go down as you get older, they tend to be maintained to a greater degree in type 1 diabetic patients. And what's most important is that the number of PDCs in these individuals, when you stimulate with a flu virus in this case, these are patients with high levels of PDCs. They produce over 500 nanograms of this is interferon alpha, and then a group of patients with a lower PDC number, and then this is the control population. So if you compare this group to this group, there's a 10-fold difference in the production of interferons by the cell populations. When we switched over to using a bioassay to detect interferons in blood, this allows for detection of one of the 13 different types of type 1 interferons instead of just one or two by ELISAs. What we found was some interesting data. So there was more interferon produced in general in type 1 diabetics as a group. This is the new onsets. But what became very interesting was if we looked at younger individuals, so kids that were between 6 and 14 years of age when we got the blood, that they produce high levels. And that was very interesting to us. If you look at when you move up in cohorts from 15 to 25, there's still some production in the patients with type 1 diabetes, but it's not as significant as it is in the younger groups. And by the time you're out to 25 years of age or older, you see less interferon production under those conditions. So this suggested that there's higher levels of interferons produced in children. So when kids get a virus infection, they probably respond with greater capacity in the interferon families as far as production and potentially response. And this may indicate why, help us understand better why diabetes tends to develop in children versus adults and adolescents. And this is just looking at some correlations for interferon levels depending on the age. And on the left is control populations where you do see this decrease. As I mentioned, there's decreased numbers of PDCs in our individuals as they age. In healthy populations, there's a lot more of them in patients with diabetes, and particularly in that young population we're looking at here, there's quite high levels of interferons that are detectable in individuals. And these are healthy kids with no active disease infection. Now, what about the genetics of this response? So we began to look at the polymorphisms in the virus receptor, the MDA5 molecule, in our IFIH1 gene. And in the pink and purple are homozygotes or heterozygotes, and the expression of these, they're enriched, generally enriched in this population of type 1 patients versus controls from what we've seen. And if you look at that young group, there's a large number that are pretty much all heterozygous. And what's interesting is there's some odd report that I've yet to understand that suggests that the heterozygote has a higher response than the homozygote to virus infections and interferon production. If we look over here at that TYK2 molecule, which again is an proximal signaling pathway of type 1 interferons and type 3 interferons, we see an enrichment here of the homozygotes in this population in general, but those that produce high levels of interferons here have these risk alleles which can contribute to diabetes. And that seems to be maintained in even those individuals that are older age. So if we characterize type 1 diabetes as a chronic inflammatory condition, and you could say that's on the basis of a multi-year inflammatory response to the islets, or you can also invoke chronic virus infections. Is there a possibility that there's a downregulation of this response over a period of time as has been seen in vitro and in a few clinical studies. And would that somehow limit the capacity of the individual to clear the infection and resolve the inflammation in the islets? So the patients have a milieu, a microenvironment, which would potentiate chronic inflammation. And is there a downregulation in that response in patients? So we looked at this by taking their peripheral blood mononuclear cells and culturing them in with or without type 1 interferons. And then we looked at the expression of 82 genes that are interferon response genes. And then we looked at the differences between controls and patients, and we broke it apart to those that had developed diabetes within a year or less, or new onsets, and then the cases that were more than a year. So does duration in this case, the duration of chronic inflammation in the islets, affect the expression in response to an interferon stimulus? And the answer was yes, it does. So patients that had disease for a longer period of time were not significantly different from controls, whereas those that had diabetes for less than a year had a significant response to the interferons. Several-fold increases here that were found in a multitude of just classical interferon response gene panels. And what about the level? Does the level of the cytokine in the peripheral blood influence the inflammation? So this is a little trickier to understand, but if you look at this legend down here, the red indicates very high levels of induction of interferon genes. And the black, low, and the intermediate is blue. And if we look at our patients here, and these are healthy controls, that what we find is, and this is the level of interferon on the y-axis. So if you look here, there's individuals that had the high levels of expression of interferons, but did not induce their interferon response genes in the same experiment I just described. So this suggested that the level of the interferons in the blood that we're detecting in a bioassay, it is suppressing the capacity of in that individual to activate interferon response genes, whereas those individuals that had low interferons down here in the bottom could induce much higher levels of interferon response. So this suggested two things: duration and the level of the interferon microenvironment, or in this case systemic environment, would modulate interferon responses. So recently, particularly in the area of cancer and chronic viral infections, there's been some really interesting research coming out about the chronicity and how it affects T-cell memory and T-cell development. And it's particularly interesting in the CD8 T-cell, which is the T-cell that has cytolytic activity and may be the major player in the destruction of beta cells in patients with type 1 diabetes. So the question we asked was, because of these chronic infections, virus infection or the chronic autoimmune response, do we see changes in altered T-cell function phenotypes and T-cell exhaustion, which is a phenomenon where T-cells after chronic stimulation go into more or less a dormant state from the standpoint of killing cancer, viruses, cells infected with viruses, and have low replicative capacity and cytokine production. So they're really kind of, it's a mechanism that the body uses to limit the expansion of inflammation and limit tissue destruction. So it's a way to put the brakes on in this case, a T-cell response. And when it gets to the point of a chronic infection, you don't want this going on forever, leading to tissue destruction and organ dysfunction. So that's what we began to look at. So this is current understanding of this area, and it's got a lot of holes to fill yet. So this is kind of a schematic of some of the papers that we've been looking at and the studies that we've been doing to kind of synthesize what we think might be happening in type 1 diabetes. So on the left-hand side, we have this description of autoimmunity, chronic, virus infection, chronic, and if you look in the cancer literature, that's a chronic inflammatory response as well. And this is all promoted by high interferon alpha and beta production, which is from some of the work of David Brooks and his colleagues. So this leads to the generation of these stem memory T cells. Now they may, as I mentioned, may come out of the you may come out of the womb with a certain component of these, and this is a population which is long-lived. These cells live in your body for years, if not your whole lifetime, and they have antigen reactivity, and so they provide a reservoir or a source of memory that allow you to gear up and take care of things like viruses. It's very interesting that if you treat these cells with PD-1 blockade, that this is the population that expands 40-fold in humans. It's also a population that probably contains autoimmune potential, in that a number of patients that are on cancer therapies where PD-1 is blocked manifest a number of autoimmune diseases as well, and this is potentially the source for that population, given that they expand and they have effector function and they contain T cell receptors, which would allow them to cause an autoimmune disease in an individual, including type 1 diabetes. So a potential bad bit of news, and potentially explaining why immunotherapies that we've used in the clinic haven't been successful, is because you can't get rid of these guys. They are there for a reason. They are there to stoke your memory. But in this case, if we have a memory to a self-antigen and they keep repopulating, the ways that we've gone about trying to modulate this disease may not be effective. It may have to be very much a long-term effect on this population, or to develop a way to totally eliminate it such that those clones of cells have been eliminated. The other way to do it, which has been suggested, is to get these cells to move into the exhausted phenotype down here, which is a cell which again has very limited antiviral or potentially autoimmune potential. And there's an investigator, Kenneth Smith, who feels that this is potentially a very good way to do this. And I won't get into all the details, but it would involve CD2 inhibition and CD4 T cell modulation. So this is what we think might be emanating, and this is based on the interferon response, the interferon production, and the genetics behind all this. So this is what we're finding, and I'll show you the data, because the data is really interesting from this standpoint. So what we found in patients with type 1 diabetes is they don't have any T cells in their islets anymore. If you look at them, in this case, you could probably hardly see it. There's 3 cells right here which label with CD45, which is a pan white blood cell marker. And when we looked at a bunch of different pancreatiform patients and controls in this case, the number of infiltrating cells in an islet is delineated in that second to the last column, where we see on the bottom in the yellow-orange color that many of these islets do not have infiltration, or if they do, it's a low number. There's a new onset patient down here, right there, that has a number of T cells that we counted in that individual. And this is after sampling many different islets. And so the range of infiltration is in that last column. So this suggested to us that there may be, in chronic disease, may be a loss of lymphocytic infiltration, a lack of interest, if you will, of the immune system into the islet responses, as well as potentially viral responses. So we took a multi-parameter approach to look at these subpopulations. There are only about 2 or 3% of the total PBMCs in the peripheral blood of the lymphocytes. And it's an 8 antibody panel, which means we're looking at 8 different proteins on the cell surface. And one of those, this is CD8 T cells, and it's gated on the 8s, as well as PD-1 levels and IL-7 receptor, and then a whole slew of other molecules which have been described for this particular stem memory T cell population. And what we see is in the blue is the healthy control population, which has a modicum of TSCMs in their peripheral blood. Type 1 diabetics, very significant levels in here. And we have within the relatives 2 groups. We have those that have a high level of these cells and those that have a low level of these cells. And so the question is, if SCMs are the source for autoreactivity, and this is a stochastic relationship, is that group on top there in the relatives the group that's going to be more prone to developing disease. What was also interesting is when we got back to the age issue. So if you look at here, the young kids, again, are closer to that y-axis. They have very high numbers of TSCMs, and this is significantly higher than controls. And if you look at the age when we looked at these individuals, they are also going down, much as we saw with the interferon production and PDC numbers. Now, what's interesting is these two dots that are at the bottom in black are individuals that develop diabetes as adults. So the numbers of SCMs that they have in their peripheral blood, and this is small numbers, highly speculative, but does this reflect risk for development of disease and when you get it? So we also went to look, there's some suggestions that the SCMs can evolve into exhausted T cells, as I mentioned. And again, we're dealing with this chronic virus and autoimmune response. And so and there's other reports in some literature, not very extensive, of exhaustion in T cells in autoimmune diseases as well. And this is looking at healthy controls. We don't really see a correlation of TSCM numbers with exhausted T cells here. But when we look at the patients with type 1 diabetes, these are established patients, we see a very significant correlation of the more TSCMs they have, with a couple of exceptions, have more exhausted T cells. So this would potentially support the evolution of autoreactive cells, if they are indeed in this TSCM population, and the generation of exhausted T cells as a result of that chronic inflammation that's occurring in response to virus or encephalitis. Now, it's also possible there's inflammation in other parts of the body going on, thyroid, etc. So this may not all be related to just the islet inflammation and the virus antiviral responses. So to summarize these, we've seen many, we've looked at pancreas as a small number, and we find that many of those insulin-positive islets have virus. And so there's still insulin being at least expressed in the beta cells of individuals with type 1 diabetes after many years, which I think is somewhat good news. We just have to figure out how to make them work. There's evidence of a chronic CVB infection and interferon expression within the context of the microenvironment of the islet. And that suggests that those in vitro studies that we did, that those interferons may be, as well as other cytokines, may be playing a role in suppressing glucose-induced insulin release. So if you treat that, get rid of the virus, block the interferons, can you reverse beta cell dysfunction? Maybe regeneration, we don't know. And then we see the systemic interferon production is most robust in younger individuals. So those kids have higher levels, and do they develop disease at an early age because their set point for interferon response or production, based on genetics most likely, is putting them at higher risk at that phase of their life versus when you're older. The genotypes may change. You may have a lower risk for inflammation in those individuals, and you develop disease more slowly and at a later age. The other interesting thing is we're looking into now, looking at the SCM population for T cell receptor expression, as well as the exhausted population. And there's some ways, without getting into detail right now, with lack of time, to the ways we might be able to manipulate some of these cells to get rid of them, or at least get them to become either a more effective antiviral response or potentially reduce their autoimmune responses. And the question is, should we be looking at kids and anyone who has in families that are looking for risk for disease? Should we be looking also not just at antibodies, but the TSCM populations as a potential for looking at the mass of autoreactive T cells that may be involved and may portend for earlier onsets? So I'll end with that, and these are the group of people that over the years have contributed to all this work. and be glad to take any questions you might have.
So, for those who are listening online, I was wondering, you'll probably have some questions. You can talk about what lessons you learned from this, in the study.
Yeah.
And, if you were to design a study, right now,
Yeah.
what we know, what would you, how would you
Well, if we have this information, and so if you look at how these chronic responses are maintained and how they may evolve from, say, a virus, this is mostly mouse data, is that the heightened inflammatory response contributes to the development of a chronic infection and the reduced ability of the immune system to clear the infection. And that's in an LCMV clone 13 mouse model, so it's not diabetes. But if you contain interferon responses in the early part of that response to the virus, you don't get this chronic evolution of T cell dysfunction. So the exhausted T cells and the SEM populations are more contained. So if you were to design a trial, because of what we saw in NIP, we saw a reduction in inflammation. We have some data from that it does suppress interferon responses as well, but we didn't do the experiments the best way to demonstrate that in NIP. But if you start early and you have these things on board, as Barry said, then having vitamin D or omega-3s at a level that are going to take down the genetically predisposed levels of heightened responsiveness or heightened production, you may be able to have an impact on disease development, slow progression, and those kinds of things. So I think it fits there, but there's also, I think, across the continuum of diabetes, which is, Game of Thrones episodes 1 through 7, that there may be opportunities there as well. And the other thing is I think that we have to think about this in the context of diabetes-related complications. There is a tendency in the mouse, and there's a tendency in humans to go and move towards chronic responses. And that's something that we can't forget when it comes to vascular atherosclerosis and retinal disease and kidney disease, wound healing and things of that nature that don't work well in patients with type 1 diabetes. Alberto?
Mike, you mentioned T cell exhaustion
Yes.
as a marker of type 1 diabetes.
Yeah.
On the other hand, there are some studies from clinical trials in which actually a CD8 T cell exhaustion was associated with response to therapy. So
Absolutely.
how does that fit together?
So the thing is the reason there are more of them there is because you have more of the precursor cells that can become exhausted. So the problem is you've got SEMs that are great things if you're trying to treat cancer, because there's some bone marrow transplants that have been done with selection of TSCMs. The reason is they don't go away. They persist for the lifetime of the transplant. And so in this case, the exhausted T cells are going to be high, principally because the TSCM population, which is their source most likely, is going to be high as well. So if they responded, and if you look at Ken Smith's papers, that's exactly what he's pushing for, is to push more of those cells into an exhausted function and phenotype so that you can, basically take away all their cytolytic activity, cytokine production and stuff. So that I think is a reasonable consideration. It's just a thing that's going to have to go on for potentially long term. Now these cells also express CD95, which means the TSCMs. And what's very interesting is that activating fast can be a problem because many cells express fast. So being able to target that to these population of cells with a combination of a T cell receptor stimulus, and the other thing that I didn't get a chance to show is we used a combo of GAD, proinsulin, and a couple of the insulin peptides, put them all together with these T cells. You can find them at the leading edge after homeostatic proliferation in culture. And we added those things in. What happened was the TSCM population moved down into an exhausted population with antigens present. Small percentage of cells, but it did reduce it did push some of those in that direction. So, and that wasn't, we don't have the tetramers yet to be able to do the studies the right way. So that's not proof that it's antigen reactive cells, but it's suggestive maybe. All right, thanks.